All right, so we're gonna finish up with the last part of kind of our introduction to genetics and Mendel's work. So if you remember, we broke this up into three parts because a lot of the short week last week we were spending to review for our summative assessment. And so today, given that we don't have a Zoom meeting and we can't really talk about much of this stuff in person till tomorrow on Tuesday, we're just going to finish up with the last smaller chunk of kind of the intro to Mendel's work in Mendelian genetics, so that tomorrow on Tuesday we can get into some more complicated stuff that addresses some of the things from the diagnostic that we weren't able to do because at the end of this unit or the first part of the genetics unit about heredity that we're going to complete here before we're done with the first quarter, we're going to learn how to do those things. So last time we talked about test crosses, and we were looking at just the basics about how to predict genotypes and phenotypes of offspring. We're going to do a lot more of that here, a little bit today, but a lot on Tuesday when we talk about laws of probability and things like that. So out of Mendel's all of his work, he came up with three laws, or three things that he saw based on his data that he collected that governed how traits were passed on or inherited. And so his first law is called the law of segregation. And what the law of segregation says is during meiosis, and we're going to talk about meiosis here a little bit later this week after we get just the basics of genetics kind of covered. Um, it says that during meiosis, during when sperm cells and egg cells are being formed, so gametes are being formed, um, one of the two copies of each gene, or one of the two alleles, go to each different daughter cell. So when we make gametes, each of the gametes has half of the genetic material as the cell that started out as the parent cell. And so those alleles segregate or they split up. So each gamete has one copy of each chromosome and therefore one copy of two alleles that each organism has for each trait. So if here was our parent cell and here are our daughter cells, okay, here is one copy of, let's say, chromosome number one, and here's the second copy of chromosome number one. Each of the daughter cells has one copy. And if these little bands represent alleles for different genes, each daughter cell has one allele for each of the gene, like maybe gene 1, gene 2, okay? They have one allele. So alleles separate or segregate in gametes. All right, and we're going to talk a little bit more about meiosis here. Like I said, after we get the basics of like just basics genetics done, because um, I like to talk about like Mendelian genetics first before we look at meiosis, because none of what Gregor Mendel discovered uh, really had to do with DNA at all, because at the point um, where he was alive and he was making his discoveries, they didn't even know about DNA or chromosomes or any of that. So it's not completely necessary to talk about that first. And I like to kind of talk about meiosis and the DNA stuff second, so you can really appreciate how much of his work didn't rely on knowing about DNA or chromosomes and things like that. So we'll kind of look at meiosis later. We'll probably talk about that more on Thursday and then put it together. So a lot of Mendel's experiments followed the inheritance of single characteristics like just flower color or just seed color. So they had to do with one trait. Flower color or seed color is a single trait. And so we call those things when we're uh, crossing or we're forming offspring from parents and looking at a single trait, we call those monohybrid crosses. Mono meaning one, so looking at one trait. Um, and other times, some of Mendel's experiments followed the inheritance of two different traits, like they crossed two parents and wanted to see what the seed color and seed shape were of the offspring. So the combination of more than one. And we called those tests dihybrid crosses because di means two. So we were looking at two traits in the offspring from a cross between parents. So we already looked kind of at the monohybrid stuff, so when he crossed white flowers with purple flowers. But this is how it might look with one of his experiments looking at two traits. So if he crossed, and this is the parent generation, a true breeding, remember that means homozygous, yellow and round pea, that means it will have, since it's true breeding, homozygous, it will have two yellow alleles, capital Y, and two round alleles, capital R. With a true breeding, that means homozygous, green, and wrinkled pea, two lowercase y's, that's the green allele, two lowercase r's, that's a wrinkled allele. And he gets an F1 generation and finds that they're all yellow round peas, all 100% of them. And then he self-crosses this F1 generation and he gets an F2 generation that has 9/16 yellow round peas, 3/16 green round peas, 3/16 yellow wrinkled peas, and 1/16 green wrinkled peas. And so now we're getting again a very specific ratio, 9:3:3:1. So this information again can tell us a little bit about how two traits are inherited at the same time in what we call a dihybrid cross. And you might remember, you might not remember that we can do Punnett squares with two and even more traits at the same time. So, what's going on here? If genes are on different chromosomes, how do they assort in the gametes? Together or independently? So, for example, if this is our cell we start off with and we're making gametes, sperm or egg cells, do we take, is this how it happens? The dominant Y and the dominant R always go together, and the recessive Y and the recessive R always go together? Or is this how it happens? Can we have any conceivable combination of Y and R? These kind of seem like the two possibilities. And what we had just seen here from his experiment, only one of them can be right, because only one of them will give us a 9:3:3:1 ratio here at the end. So let's investigate. If this first option was the way that it worked, only the two dominant alleles went together and only the two recessive alleles went together. We could predict pretty easy just using like a simple Punnett square the results. But that's not what was gotten. In this case, there is 75% yellow round peas and 25% green wrinkled peas. That doesn't look anything like the results that he got in his F2 generation. So this must not be how more than one trait separate into alleles. This is how it works, and for this we need to make a dihybrid Punnett square. And this is going to be how we're going to do that. So whenever you have two traits, you're going to, two different letters, like R, Y's and R's, right? you're going to make what's called a dihybrid Punnett square. It's going to be a big Punnett square. It's going to have 16 boxes, 4 columns, 4 rows. And what we're going to do is just like before, along the top is going to be one parent, maybe this one. Along the side is going to be another parent, maybe this one. And we're going to put above each all the combinations possible of one Y and one R we can make from that parent. So from this parent that's going along the top, we could have one dominant Y with one dominant R, one dominant Y with one recessive R, one recessive Y with one dominant R, and one recessive Y with one recessive R. So it's like the FOIL method for factoring in algebra, but it's applied to these genotypes. And since this parent is the same, like these two are the same, we get the same thing along the side. And so then each box has 4 letters, 2 Y's and 2 R's, just like the parents. And now we can see that this appropriately reflects these ratios that he found. So this is kind of the way that we're going to deal with two traits doing dihybrid Punnett squares. All right, Mendel's second law is called the law of independent assortment, which says different loci or genes separate into gametes independently. What that means is that, and let's go back here, what allele you received or the plant received for the the color allele does not influence what allele you receive for the shape allele. These two are independent. So just because you got a dominant Y doesn't really affect which allele you got for dominant or recessive for round, the wrinkled or round. And you can see that by looking at the Punnett square. Just because you got a dominant Y from this parent doesn't mean that you doesn't influence which of these other alleles you got. Sorry. You still have a 50-50 chance, even if you know you got a dominant Y allele, to get a dominant R allele or recessive R allele. So that is his laws of independent assortment. All right, there each of the different alleles for each of the different chromosomes is able to independently assort. The probability of getting one allele does not rest on the outcome of if you got a different allele or not. All right, and like I said, when we go and talk about meiosis later, we will talk about the law of independent assortment in terms of meiosis, and actually probably at the end of the week we'll talk about kind of an exception to this. All right, so this is kind of where we wanted to be at the end here today. So just looking at his law of segregation and the law of independent assortment, and then talking a little bit about monohybrids, so simple Punnett squares, little 4 square Punnett squares, and the bigger ones, the dihybrid ones. So like I said, not trying to add too much at one time, just because I know we don't have a Zoom session today, but we're going to get a lot more into this here as the week goes on. A lot of this I would imagine is probably review.